Everyone, welcome. Ian here, Nature Explored. In today's episode, I want to talk about captive breeding of the Pickergill's reed frog, Hyperoleus pickergillii, as well as where internal stakeholders as well as external stakeholders, as well as in situ and ex situ partners all took hands and worked towards one goal: to save the species. With about 5,600 known species of amphibians found around the world, and quite a number of them are critically endangered, at the brink of extinction, mostly due to the loss of habitat, but also the chytrid fungus played an enormous role, especially in Panama. But pollution, as well as predation, stress factors, climate change, all also play roles in the declining of populations around the world. But one success story is definitely the Pickergill's reed frog. It's a small little amphibian found along the east coast of South Africa, not growing more than 2.5 centimeters in total length, and that's the female. Males are much smaller. Now in this slide you can see that males are green in color and males are brown in color, with lateral lines running from the tip of the nose alongside the body to the back. But Pickergill's reed frog is only one of many reed frog species found within South Africa. So why is this one so important? Well, it is classified as endangered at this moment, but previously it was classified as critically endangered, potentially at the point of extinction. Between 2000 and 2011-12, there was 9 sites known where we could find populations of the Pickergill's reed frog. With hard work and dedication, today there's 48 known sites, potentially much more we still need to discover. Because it's an endemic species, the climate requirements are very unique and adapted to the KwaZulu-Natal province of South Africa. Now this species, as any other amphibian, plays an enormous role, a very important role to be precise, within the ecosystem and the food chain. It's not only a food source to other animals, but it also assists us with the control of insects, specifically those like the mosquito that carry malaria. KwaZulu-Natal is covered with quite a large stretch of sugarcane, but recently it all changed into development. There's still a few farms left, but development is definitely on the rise. So why the Pickergill's reed frog? If you see there, that small patch where you can find it throughout the entire South Africa is extremely small. Now it's extremely important before any species is considered to be bred in a captive environment that as much information is obtained and understood from the species. And to do that, a good way to start is to use pioneer species. In the case of the Hyperoleus pickergillii, Hyperoleus marmoratus, the common or marble painted reed frog, were used. Now, in 2006, I was still working and assisting the Johannesburg Zoo down in Gauteng, South Africa, to start up the amphibian research project. The decided species that mimics or closely resembles the Pickergill's reed frog is Hyperoleus marmoratus. And about 31 specimens were caught in 2006 in Mpumalanga district and brought to the zoo to be observed, to learn from, and to train the staff, to create protocols and standard operational procedures, to be very specific to husbandry requirements and the method how to implement this into the project. Now, it doesn't help for an ex situ partner to work alone. In situ partners is definitely a requirement because there's a lot of work that still needs to be done, as well as environmental monitoring needs to be done in the natural habitat, specifically towards the species of focus. Now, we realized after about 3 years of successful breeding of the Hyperoleus marmoratus, creating husbandry protocols, quarantine protocols for various life stages. You will see in this slide, the enclosures on the left bottom is tanks for the aquatic period or developing phase for the species. Now, eggs and embryos is the first stage. Most people think that frogs only have 4 or 5 life stages. Well, you're going to be surprised when we're going to talk about the Hyperoleus pickergill'S life stages, as there's 7 of them. But in Marmoratus' case, you will see in the right bottom corner were enclosures that got their own water purificating system installed, with 12 breeding specimens in each rack. That's one way of doing it. This is what worked at that time. But through development and further research and quite a lot of in situ studies, a BMP, a Biodiversity Management Plan, were drafted and gazetted by the South African government in 2017, stating exactly what is the path to take to work towards survival of the species, and in this case, the Pickergill's reed frog. So we need to understand the metamorphosis. We all know that amphibians means two life stages, so there's the aquatic side as well as the terrestrial side. So I'm going to explain from here on what worked for the Pickergill's reed frog in this project from 2015 to 2020. If there's any questions or concerns, please put it in the comment box below and I will try to get back to you as soon as possible. In Hyperolis marmoratus, there was one life stage less compared to the Pickergill's reed frog. The one you see here is the Pickergill's reed frog life stage development. We all start with eggs and embryos, and from there on it moves into tadpoles after 6 days and maximum 8. On day 9, we realize that there's actually a fungal growth starting, and those eggs won't hatch. After the tadpoles developed, after 2 days, they will change into developing tadpoles, changing color, starting to feed, become more active. And then later on, they will turn or change, developed into metamorphs. And then froglets will follow. And the next developing stage is young adults. They look and appear exactly like adults, except they are sexually not matured to breed and produce offspring. And then the adults. I'll take you through each of these in a moment. When you collect specimens to start, you need to ensure that your record keeping and biosecurity is accurate. You need to ensure that you can mark the specific specimens, and in this case we used VIEs, visible injectable elastomers. It will shine under ultraviolet, and we can use different colors to identify the year and location, as well as if it's a perimaterial or a captive bred specimen. This can also be used for release purposes. Biosecurity is extremely important. We use latex glove that's powder free, and all equipment from waders to spotlights, everything was brand new on the expeditions, as well as disinfectant between each expedition or activity during each expedition. Now, before you bring any specimen into a captive environment, you need to make sure that you can feed the animal the correct diet. You can create a suitable habitat that mimics its natural habitat. As well as in amphibians' case, we need to ensure that we can provide suitable enclosures that will cater for all the needs and requirements for each of the 7 developing stages. So at the left bottom corner you will see jars, transparent with screw-on lids with small air holes to allow for airflow. That's ideal for quarantine purposes, transportation, as well as for rearing. These jars is extremely easy to keep hygienically clean, and during transportation as well as for husbandry, a wet paper towel were placed at the bottom and the jars were placed on its side, sprayed twice a day with only RO water at room temperature. No other water we used, as chlorine and ammonia, all of those elements can be extremely harmful, even deadly, depending on the life stage. Even the eggs were placed inside during the embryo phase for development before the tadpoles hatched into the new life. They were then removed and placed into more suitable aquariums. On the right top corner, that is a 1 liter plastic tub that were filled up to 75% of total volume, and that marked the 100% mark. With 100% water changes required on a daily basis, the tadpoles and developing tadpole phase were all housed in that tubs. In the center you will see glass vivariums with wing doors and mesh on top were used. and the design inside was to cater for the aquatic life as well as terrestrial. The enclosure was tilted forwards at 30 degrees so that the water will accumulate in the front and it creates a suitable water or aquatic area for the frogs, while the back was terrestrial. Then leaves was placed inside, various from reeds to strelitzia leaves for them to climb, as these frogs do tend to climb at night, to high up in the reeds to find food as well as for calling when it's mating season. Lights was placed on top with UV 0.5s, and the light was strong enough to penetrate right to the bottom of the enclosure. Plastic cover were placed over the mesh roof to be opened or closed depending on the requirement. If the humidity was too high and the temperature too high, the plastic can be removed to allow more fresh airflow, or it can be closed to increase the humidity and temperature inside. Please make sure that there's no place for any amphibian to escape, irrelevant of the life stage or developing stage. Feeding is a challenge. We have studied trying to observe what food sources are available within the habitat of the Pickergill's reed frog. But during the tadpoles, it's almost impossible to study them due to the overgrown plants lying on top of the water film in the wetlands. We found it best to use spirulina as well as fish flake, finely crushed and put on top of the water film floating, while romaine blanched lettuce were placed at the bottom with pebbles that will hold it down. This method worked fantastically with Hyperolis marmoratus, but we soon realized it's not adequate for the Pickergill's reed frog, as their social structure is slightly different. They're not as competitive, but in the next episode, I'll discuss social structure and husbandry in more depth. We have removed the lettuce as we realized it's fantastic for feeding and the animals grow and there's enough food. However, it do have a limit. Muscle development is not adequate and there's a delay, as we realize that these tadpoles need to use their hind legs and tails to practice constantly, to swim and feed from the floating food, to ensure they will be able to jump and get away from predators the moment they climb out of the water and start their terrestrial life. And with the romaine lettuce, well, they tend to become quite lazy by just lying down and eating, not using the hind legs. Adult specimens walked for weeks before they were able to jump, and the muscles were strong enough. But the specimens that was raised without romaine lettuce started jumping from one side of the enclosure to the other side of the enclosure immediately. As they had no romaine lettuce, they had to exercise the muscles during the development phase. This is what it looks like without the romaine lettuce. The floating food, extremely fine, and pebbles inside so that the tadpoles can hide under. They feel more relaxed and the stress levels is definitely decreased, as well as for them to climb out if they do tend to require oxygen to breathe. As from an early stage, 2 weeks, you'll find that the tadpoles will hang motionless upside down with the mouth parts at the water full. This is an indication of lung development, where they will then start using oxygen out of the air to survive as well, and not only the gills, even though there's still about 6 weeks where they will live aquatic life before they will completely emerge into the terrestrial life. Now the frogs is collected, the permit is in place, and husbandry protocols, staff has been trained. Now it's breeding time. How do they breed? Amplexus, well known as amphibian breeding. There's 5 known methods or ways that frogs do breed. We're not talking about caecilians and toads at this point in time, or salamanders. We're focusing on the reed frogs. In the Hyperolis marmoratus case, we have seen only amplexus, where the female lay the eggs and a male will sperminate over the eggs, fertilizing them. But with Pickergill's reed frogs, we've seen 3 different ways. The one, exactly the same as marmoratus, where the male will call, the female will find a suitable male. He will fuse himself onto the female, where she will lay the eggs, and the eggs will be fertilized while the male is still on the back and she's laying it. We also noticed there was a few instances where the female laid the eggs and then went to a suitable male, where she went back to the site with the male fused on her so that he can then sperminate the eggs. Now, the last one was quite interesting. The female laid the eggs. She moved away completely, where a male found the clutch and then sperminated and fertilized the eggs without the presence of the female. Now, this could be an element of captive breeding, or it could be possible that that's actually happening in nature as well. It's extremely difficult to study in a natural environment. But there's just a few photos I've put together here where you can see eggs are laid on the leaves or even on the glass. If it's on the glass, you can use a leaf underneath, push it slightly and slowly upwards while spraying with fine water mist or RO water so the eggs roll onto the leaf, where the leaf can then be removed and placed into the jar for fine misting twice a day over the next 6 days until the tadpoles hatch. Now, it's extremely important to make sure not to disturb or change any method of husbandry during breeding season. Stress is definitely a requirement for them, because if the stress is too low, they will not breed. Also, your sex ratio needs to be accurate. We found that you need about 3 males and 1 breeding female to have success, as not all males will always breed or all females will breed. There must be competition that stimulate also breeding. The fine mist spray that is required on a daily basis in the morning and the afternoon also stimulate. The more you spray, the more it will resemble a higher humidity as well as an increase of ambient temperatures to about 24-25 degrees Celsius will automatically stimulate breeding behavior. If the eggs are laid and in the jar, 6 days later they start hatching. With about 100 eggs in the jelly-like substance as a clutch, there's quite a number of tadpoles that will hatch. But unfortunately, we've realized that an average of 69% will hatch. Not all eggs were fertilized. Now, when the tadpoles hatch, they need to get themselves strong enough to break out of the jelly substance. Now, for the 6 days of spraying that took place, you'll find that there's quite a lot of water that accumulated at the bottom of the jar. And that's adequate for the tadpoles to swim around, as they do not need food. Leave the tadpoles for a day before you remove them and place them into a small jar. How to remove them? You can siphon them with air tubing, but make sure that you boil the air tubing in hot water to sterilize it and make sure it's clean every time it's used. Also, make sure that if you do use a syringe to siphon them, that the front of the syringe is big enough or wide enough not to hurt the tadpoles when you siphon them up very slowly and place them into the jar. 100% water change is required on a daily basis, and the best idea to do that is an RO system that is filled into big 200-liter plastic drums, tested daily for chlorine and ammonia. Make sure your pH is also accurate, 6.5 to 7, neutral water. Now, development needs to take place. The tadpoles will not eat or feed on anything because they still rely on the egg sac. Now, after 2 days, you will realize they start changing color. This is when we classify them as developing tadpoles. They'll be more beige, sandy in color. They will swim faster and they will start feeding. The behavior will change completely, and there's also development that is required. So, for them to become a frog, metamorphosis will then start taking place. As I mentioned previously, from week 2, they'll start floating motionless upside down, potentially to take up oxygen. But leg development will start. So from week 5, beginning of week 5, 6, and 7, there's quite a lot of changes. The tail will turn slightly darker in color, and the tadpole will start swimming with his right-hand side towards the bottom. There's nothing wrong with him. It's actually the weight. His hind leg is busy developing and soon will be visible. 2 days later, the left rear leg will be visible. They'll swim around and use the hind legs, kicking, developing and exercising the muscles while feeding the spirulina and crushed fish flakes floating on the film of the water. At this point, make sure there's pebbles inside for them to climb out so they can also be outside the water if they do want to be. It's extremely important to maintain water temperature, and it must never go below 90. because below 19 it can be fatal. They do not require any water temperature below that. 21, 22 is usually a good temperature that produce and assist with better growth and development. Remember, it's still an ectotherm. During the changes, you will see that the tadpole will start swimming with his left-hand side towards the bottom. Now, this is, again, nothing to stress about. The tadpole is now developing his first front leg, and that will be on the left-hand side. A few days later, around about week 7 to 8, you'll find that both front legs will be visible. But at this stage, they'll stop eating as much as they used to. They'll also not be that active. The tail absorption will start. Tail do not go necrotic as people think. It's being absorbed, and they do not really require any food at this stage. Now, it's classified as a metamorph. If you look at the development I just spoke about, there's week 1, small little tadpoles. This is now the developing tadpole. If you look at the color, a tadpole will be blackish in color and even smaller. Week 2, bit of color changes. You can see organ development, long tails, and extremely active. Week 3, very similar, but you'll see the sides will also be slightly dark in color with very long tails. And you'll see the fin on the tail. Now, this is where the interesting part happens. Leg development takes place. So from week 5 onwards, it will start changing from a developing tadpole into a metamorph. Week 6 to 8, both legs and also the front legs will start to develop. Both front legs are out, it's a metamorph. Now at this stage, they can actually climb out. Make sure that there's a lid or cover and they cannot get out of this small little holding facility, as they will be able to climb out and they will die as they require water still as a semi-aquatic. Now there's a little froglet. You'll realize it looks exactly the same as the developing tadpole at week 8 as a metamorph, but the tail is absent. At that stage, they'll start focusing on food, hunting avids and crickets and bugs and in various forms, little beetles and mosquitoes. Extremely small, and male and female cannot be distinguished between each other. All of them will be beige brown in color, but they will change color. Dark in color in the evening and light in color during the day. Some of them we found to be actually bright yellowish in color. It's extremely important to maintain the records, temperatures, what's been done in the enclosure, who have done it, and remember now it's important to supplement food. Calcium powder, dusted crickets, fantastic for them to feed. Fruit flies, fantastic. They need live animals. Frogs do not prey on dead specimens. They require live food sources. Up to 5 crickets a day for an adult is adequate. 3 for a froglet is adequate for them to feed and to grow, to build up enough protein. SOPs at this stage needs to be updated. There's various life stages we discussed, and it's important to make sure that the best care and caution is taken. Until next time, take care.